Thank you very much, Dr. Yokoi Sensei. Thank you very much to the public for coming to this lecture. Thank you very much to Dr. Miyake, that he was behind me a minute ago, for having invited me to come to Japan, specifically Kyoto. I was also in Tokyo a few days ago, teaching a course and giving a talk at a symposium invited by Professor Iwata. And also thank you to my former students and now a sensei at Virginia Tech. Gota Morota has been a very good company during this time to Japan. I'm deeply honored by giving this lecture. I'm deeply honored by your presence, and specifically in this building and hall with a very important name, no less than a Nobel Prize in Physics. So I will start by making a distinction between life and abstractions. This photo is of a very famous painter called Pablo Picasso. He was originally from Catalonia, Spain, and he introduced a concept of painting, of abstract painting, that was revolutionary in art. So this photo of Picasso became a self-portrait, and that's an important thing because it relates to many of the things that I've been telling you about the history of statistical methods in animal breeding and quantitative genetics, because they're mostly abstractions. So in agricultural sciences and also in medical sciences, we deal with traits that we call them complex. That is life. What is the reason for that? Is that the reason is life is the result of a very, very large number of biochemical reactions that are mutually integrated, and they act in a concerted action together with the environment to produce outputs that in agriculture we call them phenotypes. And we measure the outputs, but not necessarily the inputs. The inputs are not always observable. So in genetics, there is a concept that is that of interaction between genes, and that is life. But we normally use abstractions. But let me elaborate what do I mean by life versus abstractions. This is a very well-known cycle in biochemistry. It's perhaps one of the first cycles that you learn. It's called the Krebs cycle, the cycle of the tricarboxylic acids. And this is a cycle in which there are 12 enzymes that intervene, which are marked with an arrow. And if you remember, there's an old paradigm in genetics, one gene, one enzyme. So that implies that this cycle is controlled by 12 genes acting together, at least, maybe less, maybe more. So the abstraction that we normally make is that every time that we change a genotype at each of these genes that call for the enzymes, you add or subtract something to the output of the cycle, but that's not true. It suffices to do a knockout of any of those 12 enzymes and the cycle that stop. That's an indication that it's interaction. So life doesn't add necessarily act by adding or subtracting from something, but rather it fundamentally based on the notion of interaction. So it must also be that the way that genes relate to phenotypes or gene products relate to phenotypes must follow some sort of nonlinear logic. And the reason is because enzyme kinetics is controlled by a nonlinear logic. We normally talk about the Michaelis-Menten theory, where the reaction rate, the speed at which a chemical reaction, biochemical reaction takes place, it depends on the concentrate. When the substrate concentration increases, the enzyme has to act faster, but it cannot increase its speed in an indefinite manner. So there is some sort of nonlinearity that is controlling the production of outputs from biochemical cycles. So because of this reason, and largely because even in spite of the revolution of molecular genetics, we still know very little about the genetics of complex traits. We tend to believe that there are many hundreds, if not thousands, of genes acting together. We tend to believe that for agricultural traits, most of the genes have a very small effect. And therefore, we try to come up with some sort of a mathematical description of the process. So, for instance, I hope I still know how to use these. The pointer is in the middle. We observe something. We postulate that there is some mathematical function that this depends on genetic variables and environmental functions. And if you think for a second, there is an infinite number of possibilities that I could come up for the form of a function. For example, here there are 5 of them, and we use actually the simplest one, which are linear representations of processes. For instance, here we could say the phenotype is a sum of genetic and environmental variables that may be independent and non-interacting. Whereas here, we could postulate that there is a joint effect of genotypes and environment that in animal and plant breeding is called genotype by environment interaction. Now, it's interesting to observe that to a large extent, the basis of quantitative genetics were set before the discovery of Mendelian rules. So let me go to a gentleman, Sir Francis Galton, that actually discovered an important principle, which is that of the regression of the offspring on parents. So this is the original data from Galton. It's a plot of the average age of parents against the average age of their children. And what he noted is that there was a linear relationship between the stature in parents and the stature of children. But when you look at groups of tall parents, they tended to produce children that on average were smaller than their parents, whereas groups of short parents on average produced children that were a little bit taller than their parents. And that phenomenon was called regression to the mean. And he thought that a linear description was a good representation of human stature from a mathematical perspective. Well, many years later, the Galton's original data were analyzed non-parametrically. So these are scatter diagrams relating the heights of fathers with sons, fathers with daughters, mothers with sons, and mothers with daughters. And using a non-parametric method, non-parametric linear regression method, they found that this line is actually not straight. It bends at some point. You see, there is some bending. And the bending perhaps may be because there is imperfect smoothing or not enough smoothing, but we still do not understand why this bend takes place, even though human stature is perhaps one of the most widely studied traits in human genetics. It's highly heritable. It has a heritability of about 80%, as estimated by many different statistical methods, but we still do not understand why this bend occurs. So this was discovered before Mendelian laws were presented, and even in the absence of an understanding of the complexities of agricultural traits, of agricultural traits. Another important statistical development that took place before the Mendelian times was that of the concept of mixture of distributions. This is a Professor Carl Pearson from Great Britain, and he was analyzing crabs from the Bay of Naples in Italy, and he measured crabs, and he found that the distribution, look at this histogram if you please, there was a bimodality. So he postulated that there were two populations, and therefore he tried to fit this histogram by overlaying two distributions, each of which would be represented with a certain probability in the overall data set. And that is today what is called a mixture of distributions, and plays an important role in statistical analysis from many different perspectives, including in whole genome prediction, where we use mixtures as prior distributions in Bayesian models. Now, after the Mendel laws became known, and I understand that Mendel laws were also discovered independently in Japan, which I learned from Dr. Gota Morota. He had that statement in his thesis, and then I double-checked, and it was right. Kanemaru, right? That was Yes. And so using Mendelian laws, there were 3 persons that actually set the foundations of modern quantitative and population genetics. Professor Sewell Wright, Professor Ronald Fisher, Wright from the United States, Fisher from the United Kingdom, and Professor John Haldane from the United Kingdom as well. Sewell Wright was interested in agriculture at the early stage of his career, but then he moved into evolutionary biology. He did most of his career at the University of Chicago, and when he was 65 years of age, he was invited by Professor Crow to move to the University of Wisconsin. So he was a retired man, an old man, and during his retirement, he wrote a 4-volume book. And he actually died at 98 years of age, and he was still reasonably well, except that he was blind, but he skidded in the ice, he slipped in the ice, he fractured his pelvis and died in the hospital. So Wright made many contributions, notoriously the concept of the inbreeding coefficient, and also an important result from a population genetic theory was the equilibrium distribution of allelic frequencies under a pure neutral and random fluctuation model. Fisher, he was trained in mathematics in Cambridge, but very interested in genetics. He set the statistical foundations of quantitative genetics, writing a paper in 1918 when he was 21 years old. That paper was rejected. And in that paper also he introduced the analysis of variance. And later on, in 1922, he wrote a paper on the theoretical foundations of statistics and introduced a method known as maximum likelihood. And Haldane was a mathematical geneticist. Actually has a paper in 1948, which I think is the first time that the notion of Bayesian methods is used in genetics. And he was using Bayesian methods to estimate the probability of a mutant in a population. Now, Wright was a very humble and gentle person. Fisher was the contrary. He was very rude, very arrogant. And Haldane was perhaps the most interesting of the 3. He was a political activist, so here he's not lecturing in genetics, he was lecturing in London to tell the workers to join the revolution against the oligarchy and the bourgeois class, because Haldane was a Marxist. He actually resigned his British citizenship, he moved to India, he became an Indian citizen, but later on in his life he returned to Britain. So in essence, these 3 people set the foundations. But we needed somebody in agriculture to put all these together in the context of animals or plants. And the person that did that was Dr. J. Lawrence Lash. And he's very famous because he developed perhaps the first textbook on scientific animal breeding. It was published in 1945 by Iowa State University Press, and he founded the famous Iowa School of Animal Breeding, and Lash probably is the professor that has the largest number of PhD students trained under him. I think he trained close to 140, 145 students. And in some sense, we are all academic great-grandchildren of Lash. My PhD supervisor, Professor Chapman, was actually the first PhD student of Chapman. So I'm a grandchild, and you are a great-grandchild of Lash. Now, Lash, I have to say, he did his PhD at the University of Wisconsin. He got his PhD in 1922, actually working in Siemens sex ratios, but then he was in Texas for a while and then moved to Iowa, and he made all these contributions. So he was able to get ideas from Wright, mostly from Wright, also from Fisher and from Haldane, and integrated them in the context of animal production and created the modern science of animal breeding. So here's a photo of Fisher, which is 1918 paper. He was very young at that time. He was an undergraduate at Cambridge University when he wrote that paper. Okay, an important concept in agriculture is that we normally select animals and plants by considering many attributes simultaneously. So today, we do what are called multivariate analysis. And a central concept in multivariate analysis is that of the association between traits measured by the coefficient of correlation. So suppose that Y1 and Y2 are measurements on two traits in animals. For example, milk production in a cow and protein composition in the milk. So this correlation may be due to either genetic associations, factors that control commonly both traits, and as well as environmental associations. And what Hazel, and also an Australian in 1936, actually a few years earlier, developed the concept of the phenotypic correlation, and this equation gives a representation of how the genetic causes and the environmental causes add up to each other, and these H squared X and H squared Y are the correlation of these two traits. So that was an important paper that set the foundations of multivariate analysis. And this is the same thing that was done in Australia by Fairfield Smith in 1936. Even though it was a plant breeding, it was published in the famous or infamous Annals of Eugenics. And he used a notion from Professor Fisher, and he called that discriminant function, but actually what he was doing is something that today we call best linear prediction, which was formalized in some sense for Henderson. So these were important precursors of multiple trait analysis, which is something that we all do in animal and plant breeding. Okay, now in the left, and I hope that this slide will not move, we have a photo of Professor Fisher when he was, of course, much older, teaching a course at Iowa State University. And this is Professor Oscar Kempthorne, also from Great Britain, but he did his academic career in the statistics department at Iowa State. I met personally Professor Kempthorne, but not Professor Fisher. And in the right, we have Professor Charles Henderson, who was here in Kyoto in 1984, invited by Dr. Sasaki. So what Henderson did was he actually generalized Fisher's ideas in the paper and made them vectorial as opposed to scalar. So Fisher's mean became a mean vector, Fisher's additive variance became a covariance matrix, and then he did many different things. He developed an algorithm to estimate the breeding value from millions and millions of animals at the same time. He extended this method to cross-sectional, longitudinal, and actually multivariate data. And then he also gave a formula for how to estimate the prediction of the breeding value of an individual that still does not exist, but it has relatives. So from the relatives, we could predict the breeding value of a cow that still is yet to be born. So at that time, we didn't know anything about markers, no genes. We didn't talk about minimum allelic frequencies, minor allelic frequencies. We didn't talk about linkage equilibrium. Nobody was too interested in causal variance because in agriculture, we felt that the causal variance of large effects would be fixed by selection, which happened to be the case. And nobody was interested in publishing Nature Genetics or National List or getting a National List of Health glands. It was a completely unglamorous thing, but yet, sorry, I have to go back. These things made tremendous contributions to the genetic improvement in production of milk, meat, fiber, energy, and resistance to diseases without any knowledge whatsoever of the molecular basis. Professor Henderson was originally from Iowa. He studied in Iowa State under Professor Hazel. He wrote his PhD thesis in 1948, and he's one of the very few persons that I met that was able to work on his PhD thesis throughout his life, because he was on linear models, and when he died, he was still working in linear models. So Henderson was instrumental. Today we talk about the N smaller than P problem. What is this? N is sample sizes, and P is the number of variants that continues growing and growing and growing due to the explosive developments in molecular genetics and in the post-genomics revolution. So we fit statistical models with many more variants and observations, and to do that you require specific methods. And I'm not going to get into the details, but an important one is called the best linear unbiased predictor, which is used in almost all species of animals for all sorts of productions to do routine genetic evaluation of animals, and Japan is not the exception. Japan also uses blob to the best of my knowledge. And this method was then found to have many different interpretations, including Bayesian interpretations, penalized maximal accuracy interpretations, spatial statistics interpretation, in even machine learning interpretations. So according to a very famous statistician from Columbia University, blob is a bit like the Holy Roman Empire. It was everywhere in the world. And Henderson was the one that started this, and it's important to remember that was the case. It's a method that originally came from animal breeding, and it postulates that in addition to the standard types of regression coefficients, we have randomly varying coefficients because they vary according to a distribution, and that produces a phenomenon called shrinkage, but that's a topic to be discussed in a course and not in a lecture because it's highly specialized. This is Professor Henderson teaching, this was in 1987, teaching a course on advances in variance component estimation, and the person to the right was a younger version of the person that is speaking to you. That's me. And I had the honor of teaching the course together with Professor Henderson. There was a Japanese student that was studying at Iowa State, which is about 7 hours away from Champaign-Urbana, Takahashi. And Mr. Takahashi would drive every Wednesdays from Iowa to Champaign-Urbana. He would take the course and then drive back. So maybe only Japanese people are able to do that. But so this is a recollection that I have. The photo is imperfect. I have a better one, but I had forgotten to put it in the presentation, and then Gota reminded me, and I downloaded from Wikipedia. I copied from Wikipedia, so the resolution is not very good. On a personal note, I have to say that I was 1 year a student with Henderson at Cornell in 1973. That year, he was not teaching his course, and even if he had taught that course, I was not ready for it because it was a very difficult course. I actually took that course when I was an assistant professor at the University of Illinois in 1978. It was one of the first courses that I took as a student, but being a professor. In animal breeding, we use data that come from a process of selection, and Henderson had a very important paper published in 1975 on how to do best linear bias prediction under a selection model. Because Henderson could not be questioned at that time, most people never questioned Henderson's paper, but actually that paper was a bit misleading in some respects. So in 1976, there was a very important paper in statistics on how to do inference from missing data. So two colleagues of mine, Sotan Im, originally from Cambodia, but he became a French citizen. He had a PhD in statistics in France. And my former student, Rohan Fernando, is a professor at Iowa State. We adapted this missing data idea and retook the selection problem from missing data perspective. And I think until these two papers and some comments that were made by Professor Robin Thompson from Edinburgh in 1979, most people did not find issues with Henderson's paper. They accepted it as a sacrosanct matter, but now there are some issues that some suggestions made that paper that probably were not the best ones, but it was an important contribution. In the middle of the 20th century, in the 50s, there was an important movement in the statistical field that started questioning the foundations of the field. In particular, what is the meaning of probability, and are we doing the best that we could do with data if we use the classical methods? So a new movement that's called the Neo-Bayes Laplace Revolution came out, and there are some very big names associated with it. James and Stein were econometricians in the west coast of the United States, Professor Lindley from University College in London, Professor George Box at the University of Wisconsin-Madison, and Professor Zellner also at the University of Wisconsin-Madison. But Zellner then was offered a job by the University of Chicago Business Schools. A professor in business school in the US made a lot more money than professor of statistics. So Zellner moved to Chicago, and he was a celebrated Bayesian statistician. So this is a photo of Bayes. His paper was published in 1763. It was a statement of conditional probability. This is a photo of Pierre-Simon de Laplace. He was a French nobleman that he liked to do applied mathematics. So he used statistical method for astronomy, for biology, but he was actually using Bayesian methods, but he called it inverse probability. And actually, Laplace had the right view, in the sense this, if you translate into English, is memory on the probability of causes as measured by the events. And that's exactly what we want to do in science. We observe things, and then we try to think about what causes the observations. Whereas classical statistics is the opposite. You postulate something that you call a model, that you think is a causal representation, and then you see with what probabilities you observe the effects. Okay, so it's exactly the opposite, but this is really what the mind wants you to do, is to measure your uncertainty about the causes once you learn the effects. The basic idea in Bayesian inference is that to construct a distribution that represents your uncertainty before you observe data. Then you observe data and represent the contribution of the data in this red curve that's called the likelihood function. And the Bayesian analysis produces a final distribution that is called the posterior distribution that is typically in between the prior and the likelihood. If the data is not very informative, then the posterior is close to the prior. If the data is very powerful, like experiments of very large scale and very good resolution, then the posterior is very close to the likelihood. So I will pass this, but it is important to note, but because the priors are subjective most of the times, then it is important to measure the extent of Bayesian learning, or said in other words, what is the influence of the prior on the posterior? Because if the prior is very influential, I could change the prior and get a different answer, and I could find the prior that gives me the answer I want to find, which of course not very scientific. Okay, I have been told many times, I don't know if this is fair or not, of being responsible for introducing Bayesian methods in animal breeding. And the story of this is that in 1982, I went to France for a sabbatical, and I started collaborating with a French animal geneticist called Jean-Louis Foulet, and we were interested in analysis of categorical data, like being alive or being dead, or different grades of a disease. And we tried to do the same thing that was what Blapp was doing, but with categorical data. So we found that the Bayesian way was one way of doing it. And then I started thinking how this could be adapted to many different problems. And I discussed this with Rohan Fernando, who was my student and colleague at the University of Illinois, and we wrote this paper, which in some respect is the introduction of Bayesian methods to animal breeding in a formal manner. And then another student of mine, Kunshan Wang, originally from China, from Nanjing, but now he's working with this pharmaceutical company in the US, Pfizer. We were the first one in the world, I think, that introduced a class of methods called Markov chain Monte Carlo, specifically Gibbs sampling. This paper was published in 1993, but it took about a couple of years to get a review and to get it published. But the first time that we presented Gibbs sampling was in 1991 in a small abstract. Okay, this methodology, Markov chain Monte Carlo, is one that allows you to do almost anything you want. So you can come up with very strange and extravagant prior distributions. This is dangerous because extravagant prior is like drinking too much before you drive, or drinking too much before you do anything. Then you combine with the data, and if your prior is very extravagant, the posterior will typically be very colorful. So you can actually fit any model, but sometimes you can get results that are, instead of reflecting the information content of the data, will reflect whatever the prior is telling you. So that's why I'm saying that it's important to measure the extent of Bayesian learning. And it was not until we started, perhaps single-handedly, to try to convince colleagues that Bayesian methods were very useful and very powerful. Then there was an explosion in genetics as soon as the Markov chain Monte Carlo methods began. So today, Bayesian methods are used for everything. And in agriculture, the epitome is what we call genomic selection, which we will talk a little bit more about it. Now, remember what I told you. In a sense, quantitative genetics started without knowing Mendelian rules. Then it was formalized and given a foundation, scientific foundation, after Mendelian rules were discovered. But it was not until molecular markers came into the picture that you could actually start dealing with things that the theory was telling us. So I want to mention the important contributions of the school from the University of Edinburgh. In particular, this is Douglas Faulkner. Is the author of a very classical book in quantitative genetics, Introduction to Quantitative Genetics. I met him personally in Edinburgh in 1982 and then in 1998. I don't know if, he was alive still in 1998, but I met him in 1982. Very important book. And then two persons, Alan Robertson and Bill Hill. Professor Robertson passed away in 1987. He was very interested in using ideas of no less than Moto Kimura to arrive at the probability of fixation of an allele in a small population that was undergoing selection. So Robertson has a number of important papers on this matter. And this is Professor William Hill. He's probably getting close to his 80s. He's fortunately still with us. He's still alive, but he has retired. And maybe he's a bit less active now than he was a couple of years ago. But he made many contributions, including all a range of papers on linked disequilibrium. That is something that became quite important in the search for quantitative trait loci. William Hill is a person that I admired very deeply. He was always very influential in my thinking. I always respected a great deal what he said, and perhaps it's come because I had to present a paper as a student in 1972, and I was able to derive, I was able to present all the formula, but I was never able to derive many of them, and I still can't. Then markers came into the picture as a consequence of the mapping projects. And you know what we have done, a lot of developments, and we have most species have full sequences, and the cost of sequences coming down and down and down and down. So the basic idea that came is that of relating variation at some loci called singular nucleotide polymorphisms and try to relate variation at the level of markers with variation at the levels of the phenotype. So, for instance, there are markers encoding regions and regulatory sequences elsewhere, and the idea was to relate markers to phenotypic variation to see if they could be used either for agricultural breeding or perhaps for clinical practice in personalized medicine. So the first type of paradigm, which is very active in human genetics, is called genome-wide association studies, where they search for genomic regions that are associated with traits. The first study was done in Japan with macular degeneration. There are many recognized genome-wide association studies, and if you follow some of the papers like PLOS Genetics, Nature Genetics, Nature Review Genetics, there is a great deal of emphasis, and there are some, they measure everything, especially diseases, but sometimes there are some traits that you may think are humorous, like how hard you shake the hands of others. Maybe in Japan is the angle with which you bow, the propensity to cry, and many other things. There is a real question as to what extent genome-wide association studies are helpful for practical purposes, both in agriculture and in medicine, but it's still a very active field. So this is a standard way of representing genome-wide association studies. Each of these points is a chromosome by chromosome, marker by marker scan of an association. Normally regression methods, linear or logistics are used. And because you do many scans, hundreds of thousands, you have to adjust for multiple comparisons. There's a problem called multiple testing. So there are very stringent significant thresholds, and when this threshold is exceeded, then you declare a significant association. Whether that association has a predictive value or not is a very doubtful issue, because statistical significance and prediction are not necessarily related, okay? Sometimes statistical significance lead to better predictions, sometimes it doesn't, and sometimes predictions are better using things that are not statistically significant. This is something that we know quite well in animal and plant breeding, but perhaps was not that well known to other scientists. And the funny thing about this is that this paper published in the Proceedings of the National Academy of Sciences, the senior author came from a Department of Political Science. And frankly, the political scientists are not very good at predicting things. Just think about the elections in the United States in 2016. Nobody was able to predict that Donald Trump would become president of the United States. So there is a second paradigm that has become very dominant in animal and plant breeding, that is called whole genome regressions. The paper was presented, published in 2001, and the idea is, instead of looking at marker by marker thing in the model, like trying to find QTLs, just use all the available variants that you have and put them all together in a model, and then use methods to temper this problem that are called P larger than N. There are methods to avoid overfitting and to convert these methods into good prediction machines. So genomic selection was first applied in dairy cattle in the United States and in the Netherlands in 2007, and now it's applied in dairy animals, beef animals, sheep, pigs, aquaculture, maize, barley, wheat, coffee, sugarcane, palm oil, rubber trees, forestry, etc. So it has become a very important and useful technique. So These papers suggested 3 methods, but then many people have extended the class of methods, which include many, many different methods of linear regression, and more recently, methods of machine learning, and we will say a little bit about it later on. So to give you an idea how genomic selection works, you have to have some chip. In dairy cattle, now we are using chips of about 800,000 markers, and the number of animals that were genotyped in this paper was published in 2017, so the data are only up to 2016. At that point, we had a bit close to 200,000 bulls genotyped in the US and about 1.2 million cows in the Holstein breed only. So now I wouldn't be surprised if about half of the cows in the US are genotyped, and we have about 9 million cows, maybe a bit more. In another paper, they estimated using genomics what have been the trends in these populations. So I want to bring to your attention the blue lines. This is the estimated genomic trend for milk production, this is for fat production, and this is for protein production. And genomic selection began to be applied in about 2007, so you see that a couple of years after the introduction of genomic selection, then the population started changing much more rapidly than before. And this is due to 2 reasons. One is because the predictions based on markers are a bit more accurate than pedigree-based prediction, but also because you can get estimates of the breeding value of animals much earlier in life, so you can make selection decisions and choose your replacement bulls, for example, much earlier than before. Under standard progeny testing, the generation interval was about 6 years, and now the generation interval is about a little bit less than 2 years in cattle. So we are turning generations much more rapidly. And in the right, we have what are called functional traits. This panel is the somatic cell count in milk. The smaller the number of cells, the healthier the cows are. So we are going in the right direction. This is the productive life, namely the number of days that a cow stays in the herd. So the cows now have a larger productive life, genetically speaking, and this is reproduction. In the Holstein breed, which is actually a machine of producing water, reproductive performance was deteriorating very severely, and the reason was that increased selection for milk production was making the animals less functional. So then when selection objectives changed to give more emphasis to reproduction and genomics came into the picture, then things started moving in the right direction. So it looks like we are doing things right, at least in dairy cattle, getting more productive animals that are healthier, that they reproduce better, and that they stay for a longer period of time in the life. One problem, but we still do not know how to cope with, is that of gene by gene interactions, actually gene by gene by gene by gene. The medical geneticists talk about gene by gene interaction, and I think it's a bit misleading because, as I said before, from a biological perspective, high order interaction might be quite important. However, when you use statistical methods, you do not find much evidence of interaction. So the question is it because the methods are wrong, or is it because interaction is not important? Well, the answer, if there are, of course, many groups in the world that try to make sense about interaction by using these so-called systems approaches or structure or systems biology. This is one representation. So some people are repeating something that used to be said in the mid-60s in anthropology, and in Germany there was the theory of general system theory of Ludwig von Wernher-Lampe. They have to take a look at the system as a whole and not at the parts. So some people are trying to use this systems approach, but once you come up with a diagram like this, it's very, very difficult to make sense out of it, even though there are just a few nodes in this system that in some sense are representing something related to obesity or hypertension or something like that. Now, This problem of why interaction doesn't show up statistically was investigated in a paper by William Hill, and they actually found that even for epistatic systems, most of the variance turns to be additive. So in a sense, these important interactions from a biological point of view, they are hiding. They are generating additive variance, so we will never be able to understand complexity using linear models, because linear models or standard parametric method most of the times will tell that interaction is not important or that it contributes very little to the variance. Yet, we know that mechanistically it's expected to be quite important. So what's happening statistically? Well, what happens, if you remember your basic mathematics, in calculus, there is something called the Taylor series expansion of a function. So you have a function, you expand it by using a linear term, a quadratic term, a cubic term, an nth order term, and quite often, near this point of the expansion, the linear function does a very good job, and that is essentially what's happening. The linear model, this additive model, is giving you a representation that it locally is quite good, is helpful to do predictions for a reasonable, in a neighborhood of time that is reasonably small, but then when things change, you have to adapt your regression line. But we still do not know all the surfaces, concavities, and convexities of the genotype to phenotype map because there are just far too many dimensions. So sorry about that. The fonts do not embed very well. So in essence, if you believe the statisticians are what comes out of most quantitative genetic analysis, then life is a straight line. And it's an operational thing. You go out to the street here and you look in this direction, that direction, and the earth looks flat, but it's not flat. So what I'm trying to convey here is the statistical methods are actually good to describe, good to predict, but not always good to explain. Just think about Galton. We still do not know why this regression bends at some point, which is about 67 inches in humans. So if we want to understand something like this, you have to cross that, okay? To understand something like this, there are better approaches, molecular genetics, for example. But of course, you have to reduce the system. You'll be very strongly reductionist. But given resources, given money, given time, sometimes you are able to decipher or understand important genes, such as a TP53 gene, which is one of the first battles of defense against cancer in humans. So people talk about genetic architecture statistically. Well, these are some beautiful piece of architecture. This is an Iraqi architect that did most of her career in London. This is an Uruguayan architect that constructed this bridge that is very beautiful when you look at it from an airplane. But if you go to Tokyo and you go to the Tokyo International Forum, which is a magnificent building, especially Hall G, it was done by this person that happens to be born in the same country where I was born, which is Uruguay. This is the famous Bilbao Museum by Frank Gehry, and this is the scientific city of Santiago Calatrava in Valencia, Spain. So this is architecture, and not how many bricks, how many steel, and how much hours of human labor it took. Architecture is the integration of all these things, and the same applies to genetics. Okay, so the bricks and the hours of labor and the kilos of steel or tons of steel are the linear regression method, but the architecture is something much more subtle. So for that reason, I do not think that we'll ever be able to understand the genetic architecture of complex traits, but we have to regard the method that we have in a modest manner. They can predict, they can describe, but they are not very good to explain. So many people actually accompany me in this particular perspective. And one clear case is the application to try to get data of almost anything and then try to make sense out of it. And we know, we interact with the computers, we enter in Google, we go to Facebook, and they are learning from you. And actually, when you do an information retrieval, what comes back in for example, in Google, is something is really very relevant to what you're looking. So there are ways of learning about not necessarily understanding, but of learning and predicting and classifying that does not require a theory that involves parameters or gene frequencies or linkage equilibrium, etc. And this is the area of machine learning. So this is a common statement in the US: if you cannot beat them, join them. So if you do not understand complex data, that doesn't mean that you have to give up. There may be methods that allow us to emulate how complex data behave without perhaps understanding very well why they do so. And these are many of the approaches that are used in machine learning, such as neural networks, universal approximators, kernel methods, non-parametric methods, support vector machine, random forests, and animal and plant breeders have actually done contributions to it without knowing. Galton, doing data mining, the groups that deal with sampling methods, in sampling, cross-validation in animal and plant breeding, kernel methods, Bayesian networks. Dr. Morota used a Bayesian network to study linkage equilibrium, for example. Methods that use meta methods like boosting, bagging, support vector machines, etc. So we have been doing methods without knowing, excuse me. And actually, best linear bias predictor, as I said before, is a linear neural network, which is considered to be a universal approximator. Another class of method is based on non-parametric regression. It's trying to find a function that is best in a Hilbert spaces of function, which is a very vast space of functions. These methods were actually developed, the idea of using this method came from our group. The first application was done in chickens by one of my postdoc, Oscar Gonzalez Recio from Spain. And Gota did also work with reproducing current Hilbert spaces regression. There are some recent papers in which they have used this idea to examine to what extent accommodating genotype-environment interaction enhances prediction. So there is a paper by a couple of Italians that work in North Carolina State, and also by Diego Jarquin, that he was a colleague of Dr. Morota at the University of Nebraska, and it seems to give considerably better predictions in some cases for dairy animals, especially for young bulls. Now, with the question of that animal production is becoming less and less popular, and there is concern about environmental contamination and especially in the case of ruminants, contribution of methane to global warming, there is a great deal of interest in learning about the interaction between your metagenome, all the bacteria and viruses live within you, and for that we don't have any theory whatsoever. So machine learning algorithms have been suggested as a way of trying to do something with it. The most recent generation of methods actually comes from the field of artificial intelligence. I don't know if you are familiar with this book writer called Dan Brown. He wrote some very popular books. These are fiction, the Da Vinci Code, Angel and Demons, Inferno. And this is a book, it's based on a plot to kill the King of Spain. And the central character in the book is a computer. It's called Winston, like Winston Churchill. And it uses a method called deep learning that you probably have heard about. So deep learning is something that it's not very new in science, and we have actually done a bit of deep learning without knowing it. This is It's a neural network. It's a mathematical representation of relationship between inputs and outputs. And in the middle, you have some cells that do mathematical operations that are called neurons, and the deep learners are just neural networks with several layers like this, multiple layer neural networks. But there is a new class of algorithms that have been suggested that similarly are very good for doing many things, like image analysis, consumer choice. For instance, when you try to buy a TV, and you go to a search machine and you specify things, that normally a good choice will be made for you. So these are the deep learners, and the question came that, well, maybe we should abandon all the methods that we are using for predicting performance in animals and humans and move to deep learning. So let's see what evidence do we have that deep learners can help us in artificial breeding of animal and plants. There have been many studies, but I'm going to concentrate on some ones, and these are not terribly biased to show the negative performance of deep learning. This is a paper from Indonesia. They look at 8 maize traits, their flowering, grey yield, etc., and they compare 4 methods: best linear bias prediction, a Bayesian method called the Bayesian lasso, this non-parametric method called reproducing kernel spaces regression, and this is a deep learner. It's called a deep belief network. These are correlations, so the larger the better, the closer they get to 1 the better, and observe that in the 8 comparisons, deep learning was better only in 2 out of the 8 comparisons, and perhaps not significantly better, because for instance, if you compare this number with this number, there's a difference in the 3rd decimal level. The same happens here, but in 5 out of the 8 comparisons, the deep learner was the worst. Okay, and if you listen to what comes in the newspapers all the day and what the computer scientists tell you, we should move into deep learning. Well, according to the paper, not yet. So then a study in which I was more an instigator than a significant contributor, they compared deep learning, which is the red, with GBLUP for several wheat traits. So there are 9 data sets, including data from Iran. This is a situation in which the deep learner was always worse than GBLUP, not only in Mexico, but also in Iran. Now, here there is another class of models. Here there was interaction, including the model, which is important in plant breeding. But if you omitted interaction, then there were a few instances in which deep learner was better than GBLUP. But there are many more instances in which that was not the case. So we cannot conclude that we should replace our battery of methods by deep learners. And every prediction problem is unique. So it's difficult to come up with universal recommendation. This is another study by the same group, but now you see multi-trait, multi-environment deep learning models. And again, these are 2 Bayesian methods. These are 2 deep learners. Observe that there is really not any clear, for example, with Iranian data, the deep learner were much worse than the parametric methods, and there are really no clear differences at all in favor of deep learner. The same has been found with humans. This is stature in humans. The pink and the magenta are the deep learners. The green ones are standard multi-layer perceptrons that are neural networks, but without using the convolutional neural network algorithm. And the first 2, the gray and the black, are base B, which is a linear regression, and this is genomic plot. So for stature, no evidence that deep learning was better, but for bone hill mineral density, this is in humans, the deep learners were sometimes a total disaster, like here. See? These are different ways of fitting SNPs in the model. So we don't have any evidence that deep learning is always the best, and actually we do have evidence that they can be very unstable. Whether that is the result of that they have done things wrong, or maybe that everybody uses the same software, is a matter to be decided. But as of today, we do not have any clear evidence of the role that deep learning could play in animal and plant breeding. Well, I am reaching the conclusion. I'm sorry it has taken me more than an hour. So if we look at retrospectively to this historical narrative, and I have to say, like when you read history, the narrative is typically very personal and very biased. This is a personal narrative of history. Perhaps if you had taken somebody else of my age and my experience, the history would have been different. But this is the history as I view it, okay? I think it is fair to say that animal breeders have incorporated the statistical idea very rapidly. Deep learning came up and it was tried right away. They have also contributed to statistics as well by contributing methods such as BLUP, REML, Bayesian modeling, Bayesian linear regression, genome-enabled prediction. Something that we do know very well is that inference and prediction are very different. They are connected, but not the same thing. So sometimes if you learn the state of the world too well, and you understand everything, but the model is very, very complicated, the prediction may be very poor, because the parameter of the model cannot be estimated effectively. I think it's fair to say that the emphasis in GWAS, which is really excessive, there are tens of thousands of papers in GWAS, that many of these will be GWAShed away simply because the standard GWAS model is a very poor description of complexity. So you cannot, when you estimate a single regression coefficient, and there are many other variables that have been ignored in the model, the results are bound to be biased and are bound to be misleading. What about, here there is a mistake in the slide. These two statements should have been separate. Kernel-based methods have been found quite useful in prediction. Actually, if I had to choose between a deep learner and a kernel method to predict something, I would probably try first a kernel method because I think it will be more stable. Systems approaches, well, there is always this big discussion that these nice systems approaches can be used with small systems with experimental organisms such as Drosophila or Arabidopsis or Caenorhabditis. But then when you extrapolate, you have to remember that pigs do not fly yet. Not even cars fly yet. So there are challenges to parametric methods posed by this massive amount of information. So many groups in animal and plant production are moving to other ways of exploiting massive data and also other ways of looking at how you use this massive data to come up with predictions. And Dr. Morota is one of the pioneers of these new generations. So an example where this may be useful is what's called genetic metagenomics. So it's what we talk about metagenomics and like to learn, for example, the composition of microbiota, but there may be genetic variability for composition of microbiota. So then we have two layers, the genetic layer and the metagenomic layer. How to put that together in a model is something that is a topic for investigation. So are we moving in a direction where everything will be semi-parametric, data guided, and machine learning? I do not know. I truly do not know. But I'll just finish with the following comment. A couple of years ago, the famous British magazine, The Economist, said that the dairy farm of the future will just have two employees, a man and a dog. And the job of the dog will be to chase out people that come to touch the robots. Okay? So if you touch the robot, the dog will come and bite you and chase you away. And the role of the man will be to feed the dog. Okay? So we may be moving in that direction, which will change the landscape as we know, but I don't know to what extent this is an unavoidable matter. Well, I have reached the conclusion. I would like to thank you for your attention. I'm sorry that I went over time. I tend to indulge into digressions, and today was not an exception. I would like again thank Dr. Nokoi, right? And Dr. Miyake for the invitation to come to Kyoto. It has been a real pleasure, both for my wife and I, to be in Japan. We have found you people extremely hospitable and courteous, and it is just our inability of learning Japanese that puts us in trouble, because everybody comes and tries to help us as best as we can. So thank you very much to all. Arigatou gozaimasu.